An RNA virus is a virus that has RNA as its genetic material. This nucleic acid is usually single-stranded RNA but may be double-stranded RNA. Notable human diseases caused by RNA viruses include Ebola, hemorrhoragic fever, SARS, the common cold, influenza, hepatitis C, West Nile fever, polio, and measles. The ICTV classifies RNA viruses as those that belong to Group 3, Group 4 or Group 5 of the Baltimore classification system of classifying viruses and does not consider viruses with DNA intermediates in their life cycle as RNA viruses. Viruses with RNA as the genetic material but that include DNA intermediates in the replication cycle are called retroviruses, and comprise Group 6 of the Baltimore classification. Notable human retroviruses include HIV-1 and HIV-2, the cause of the disease AIDS. Another term for RNA viruses that explicitly excludes retroviruses is ribovirus. Characteristics: single-stranded RNA viruses and RNA sense. RNA viruses can be further classified according to the sense polarity of their RNA into negative sense and positive sense, or ambisense RNA viruses. Positive sense viral RNA is similar to mRNA and thus can be immediately translated by the host cell. Negative sense viral RNA is complementary to mRNA and thus must be converted to positive sense RNA by an RNA polymerase before translation. As such, purified RNA of a positive sense virus can directly cause infection, though it may be less infectious than the whole virus particle. Purified RNA of a negative sense virus is not infectious by itself as it needs to be transcribed into positive sense RNA. Each variant can be transcribed to several positive sense RNAs. Ambisense RNA viruses resemble negative sense RNA viruses, except they also translate genes from the positive strand. Double-stranded RNA viruses. The double-stranded RNA viruses represent a diverse group of viruses that vary widely in host range, genome segment number, and virion organization. Members of this group include the rotaviruses, renowned globally as the most common cause of gastroenteritis in young children, and picoburnaviruses, renowned worldwide as the most commonly occurring virus in fecal samples of both humans and animals with or without signs of diarrhea. Picoburnaviruses have also been recently reported in respiratory tract samples of pigs, and blue tongue virus, an economically important pathogen of cattle and sheep. In recent years, remarkable progress has been made in determining, at atomic and subnanometeric levels, the structures of a number of key viral proteins and of the virion capsids of several dsRNA viruses, highlighting the significant parallels in the structure and replicative processes of many of these viruses. Mutation rates. RNA viruses generally have very high mutation rates compared to DNA viruses, because viral RNA polymerases lack the proofreading ability of DNA polymerases. This is one reason why it is difficult to make effective vaccines to prevent diseases caused by RNA viruses. Retroviruses also have a high mutation rate even though their DNA intermediate integrates into the host genome, because errors during reverse transcription are embedded into both strands of DNA before integration. Some genes of RNA virus are important to the viral replication cycles and mutations are not tolerated. For example, the region of the hepatitis C virus genome that encodes the core protein is highly conserved, because it contains an RNA structure involved in an internal ribosome entry site. Replication. Animal RNA viruses are classified into 3 distinct groups depending on the genome and mode of replication. Double-stranded RNA viruses contain from 1 to a dozen different RNA molecules, each coding for one or more viral proteins. Positive-sense ssRNA viruses have the genome directly utilized as if it were mRNA, with host ribosomes translating it into a single protein that is modified by host and viral proteins to form the various proteins needed for replication. One of these includes RNA-dependent RNA polymerase, which copies the viral RNA to form a double-stranded replicative form. In turn, this directs the formation of new variants. Negative-sense ssRNA viruses must have the genome copied by an RNA replicase to form positive-sense RNA. This means that the virus must bring along with it the RNA replicase enzyme. The positive-sense RNA molecule then acts as viral mRNA, which is translated into proteins by the host ribosomes. The resultant protein goes on to direct the synthesis of new variants, such as capsid proteins and RNA replicase, which is used to produce new negative-sense RNA molecules. Retroviruses have a single-stranded RNA genome but, in general, are not considered RNA viruses because they use DNA intermediates to replicate. Reverse transcriptase, a viral enzyme that comes from the virus itself after it is uncoated, converts the viral RNA into a complementary strand of DNA, which is copied to produce a double-stranded molecule of viral DNA. After this DNA is integrated into the host genome using the viral enzyme integrase, expression of the encoded genes may lead to the formation of new variants. Classification. Classification of the RNA viruses has proven to be a difficult problem. This is in part due to the high mutation rates these genomes undergo. Classification is based principally on the type of genome and gene number and organization. Currently there are 5 orders and 47 families of RNA viruses recognized. There are also many unassigned species and genera, related to but distinct from the RNA viruses of the viroids and the RNA satellite viruses. These are not currently classified as RNA viruses and are described on their own pages. Positive-strand RNA viruses. This is the single largest group of RNA viruses with 30 families. Attempts have been made to group these families in higher orders. These proposals were based on an analysis of the RNA polymerases and are still under consideration. To date, the suggestions proposed have not been broadly accepted because of doubts over the suitability of a single gene to determine the taxonomy of the clade. The proposed classification of positive-strand RNA viruses is based on the RNA-dependent RNA polymerase. 3 groups have been recognized: biomaviruses, commaviruses, nepaviruses, nodaviruses, picornaviruses, potiviruses, sobomaviruses and a subset of luteoviruses, the picorna-like group. 2. Calmaviruses, dianthoviruses, flaviviruses, pestiviruses, tombus viruses, single-stranded RNA bacteriophages, hepatitis C virus and a subset of luteoviruses, the flavi-like group. 3. Alphaviruses, carlaviruses, furoviruses, hordeviruses, bateviruses, rubaviruses, tobraviruses, tricornaviruses, timoviruses, apple chlorotic leaf spot virus, wheat yellows virus and hepatitis E virus, the alpha-like group. A division of the alpha-like supergroup on the basis of a novel domain located near the N-termini of the proteins involved in viral replication has been proposed. The two groups proposed are the alphaviruses group and the typoviruses group. The alpha-like supergroup can be further divided into three clades: the ruby-like, tabamo-like, and timo-like viruses. Additional work has identified 5 groups of positive stranded RNA viruses containing 4, 3, 3, 3, and 1 order, respectively. These 14 orders contain 31 virus families and 48 genera. This analysis suggests that alphaviruses and flavoviruses can be separated into 2 families, the Togaviridae and Flaviridae, respectively, but suggests that other taxonomic assignments, such as the pestiviruses, hepatitis C virus, rubeviruses, hepatitis E virus, and arteriviruses, may be incorrect. The coronaviruses and toroviruses appear to be distinct families in distinct orders and not distinct genera of the same family as currently classified. The luteoviruses appear to be 2 families rather than 1, and apple chlorotic leaf spot virus appears not to be a clusterovirus but a new genus of the Patexviridae. Evolution. The evolution of the picornaviruses based on an analysis of their RNA polymerases and helicases appears to date to the divergence of the eukaryotes. The putative ancestors include the bacterial group 2 retroelements, the family of HTRA proteases and DNA bacteriophages, double-stranded RNA viruses. This analysis also suggests that the dsRNA viruses are not closely related to each other but instead belong to 4 additional classes: Bernaviridae, Cystaviridae, Partitaviridae, and Reoviridae, and 1 additional order of one of the classes of positive ssRNA viruses in the same subphylum as the positive strand RNA viruses. One study has suggested that there are 2 large clades. One includes the Calliciviridae, Flaviviridae, and Picornaviridae families, and a second that includes the alpha-tetraviridae, Bernaviridae and Cystaviridae, Nodaviridae, and Permutotetraviridae families. Group 3, dsRNA viruses. There are 11 families and a number of unassigned genera and species recognized in this group. Family Amalgaviridae, Family Bernaviridae, Family Chrysiviridae, Family Cystaviridae, Family Ornaviridae, Family Hypoviridae, Family Megabernaviridae, Family Partitaviridae, Family Picobernaviridae, Family Reoviridae includes rotavirus. Family Totiviridae, Family Quadriviridae, Cucurbit yellows associated virus, Sclerotinia sclerotiorans debilitation associated virus, Spitistolus festinus virus 1. Group 4, positive sense ssRNA viruses. There are 3 orders and 33 families recognized in this group. In addition, there are a number of unclassified species and genera. Order Nidoviridae, Family Arteriviridae, Family Coronaviridae includes coronavirus, SARS family Mesonaviridae, Family Rnaviridae. Order Picornaviridae, Family Dysastroviridae, Family Iflaviridae, Family Marnaviridae. Family Picornaviridae includes poliovirus, rhinovirus, hepatitis A virus. Family Cucurbitae includes subfamily Comuviridae, genus Bacillariornavirus, genus Dissipavirus, genus Labernavirus, genus Sequiviridae, species Kelpfly virus. Order Timovirales, Family Alpha Flexiviridae, Family Beta Flexiviridae, Family Gamma Flexiviridae, Family Timaviridae. Unassigned Family Alpha Tetraviridae, Family Alvanaviridae, Family Astroviridae, Family Barnaviridae. family Beniviridae, family Bromoviridae, family Calliciviridae, includes Norwalk virus, family Camotetraviridae, family Closteroviridae, family Flaviviridae, includes yellow fever virus, West Nile virus, hepatitis C virus, dengue fever virus, Zika virus, family Fusariviridae, family Hepaviridae, family Leviviridae, family Luteoviridae, includes barley yellow dwarf virus, family Nonaviridae, family Nodaviridae, family Permutotetraviridae, family Potiviridae, family Togaviridae, includes rubella virus, Ross River virus, Syndbus virus, chikungunya virus, family Tombosviridae, family Virgaviridae, unassigned genera, genus Bluna virus, genus Celevirus, genus Hygrivirus, genus Ideovirus, genus Negevirus, genus Aumavirus, genus Polemavirus, genus Sobelmavirus, unassigned species Asiathosiphon Possum virus, blueberry necrotic ring blotch virus, Chara australis virus, extra small virus, Jingman tick virus, LeBlanc virus, Orsay virus, Plasmopara halsteadi virus, Santoi virus, Solenopsis invicta virus, 3 satellite viruses, chronic B paralysis associated satellite virus. Group 5 Negative sense ssRNA viruses. This groups consist of 12 families recognized and a number of unassigned genera and species. Order Mononegavirales. Family Bornaviridae includes Borna disease virus. Family Filaviridae includes Ebola virus. Marburg virus. Family Myrmonaviridae. Family Nyamaviridae. Family Paramyxoviridae includes measles virus, mumps virus, Nipah virus, Hendra virus, and Newcastle disease virus. Family Pneumaviridae includes human respiratory syncytial virus. Family Rhabdoviridae includes rabies virus. Family Sunviridae. Unassigned mononegaviral genus Amphivirus. Unassigned mononegaviral genus Arlavirus. Unassigned mononegaviral genus Schenkti-virus. Unassigned mononegaviral genus Crustavirus. Unassigned mononegaviral genus Waste-Dry-virus. Unassigned families. Family Arenaviridae includes Lassa-virus. Family Bunyaviridae includes Hantavirus. Crimean Congo hemorrhagic fever. Family Ophiviridae. Family Orthomyxoviridae includes influenza viruses. Unassigned genera. Genus Delta virus includes hepatitis D virus. Genus Tenua virus. Genus Emara virus. Source